Today, we shall try to understand the processes involved in the function of vision, that is, the physiology of vision. Vision, or the ability to see, is the primary function of the two eyes. It is achieved by the eyes, the visual pathways, and the visual cortex, and is fine-tuned by the extraocular muscles and intraocular reflexes. Let us have a deeper look at the function of vision. Vision is not only the ability to see, but it allows innumerable combinations and permutations like seeing in varying background light, dark hues and shades, at various distances, perceiving various colors and shades, different levels of illumination. It also allows perceiving texture, contour, depth, motion and speed. So, if you look at it, vision itself comprehensively comprises of various finer things that we can perceive. The physiological processes involved in vision are image formation, impulse generation, impulse transmission, perception, and fine-tuning. The image formation is facilitated by the passage of light rays through the ocular media and refraction that allows the light rays to focus on the retinal receptors. The retinal receptors receive the light rays and generate the visual impulse, which is transmitted through the visual pathways and perceived at the level of the visual cortex. Fine-tuning is achieved by various extraocular and intraocular reflexes. The pupils, the accommodation, the binocular reflexes, and ocular motility. This figure shows the media and the path of the light rays. Media is comprised of the structures through which the light rays pass, and these are the precorneal tear film, the cornea, the aqueous humor, the lens, and the vitreous. The light rays, they fall on the cornea and undergo refraction at the level of the cornea and the lens to get focused on the retina. We have seen that from anterior to posterior, the media comprise of the tear film, cornea, aqueous, crystalline lens, and the vitreous. And the major factors responsible for maintaining transparency of the media are their anatomical structure, avascularity, and adequate hydration. From refraction of light rays to visual perception, the main mechanisms that are involved are phototransduction or initiation of visual impulse. This is a function of photoreceptors, the rods and the cones. Processing or transmission of visual sensation, which is a function of retina and visual pathways, and visual perception, which is a function of the cerebral cortex. Phototransduction or initiation of the visual impulse is a function of photoreceptors, the rods and cones. Just like the different sensation-specific receptors in the skin, stimulation of the rods and cones in the retina causes a visual impulse. Rays falling on the optic disc where there are no receptors therefore do not result in a visual sensation. In the visual field, this area is represented by a non-seeing area called the blind spot of Marriott, as seen in figure 2. Light falling on the rods or cones triggers a series of photochemical and electrical reactions. This figure shows the uniocular and the binocular fields and the hill of the vision. This is the field of the left eye and this is the field of the right eye, the central parts corresponding to the fovea. And when both the eyes are working together, then there is a binocular field and there is a uniocular field for the right eye and a uniocular field for the left eye. This figure shows the hill of the vision. The apex is the best visual acuity, which is represented by the fovea, and as we move away from the fovea, there is a gradual decline in the visual acuity. On the temporal side, there is a gradual decline and there is a sudden dip to a non-seeing area. This dip corresponds to the blind spot or the optic nerve head where there are no visual receptors. The blind spot is represented in the visual field on the temporal side of the fovea here as a black spot in the right eye, and this is in the left eye. The light stimulus initiates changes in pigments of rods and cones. The most understood pigment, rhodopsin or visual purple, is found in abundance in the rods. Photochemical changes are a cycle of rhodopsin bleaching stimulated by the light and regeneration facilitated by vitamin A. There is a state of equilibrium between these two processes of bleaching and regeneration. This is called the visual cycle. 3 different pigments are associated with the cones. Each reacts preferentially to different bands of wavelengths of light. Rhodopsin is a chromoprotein consisting of a reactive part, the chromophore, attached to a supportive protein, the opsin. Chromophore is a carotenoid, vitamin A aldehyde or 11-cis-retinal, responsible for preferential absorption of light around 500 nanometers. On exposure to light, there is photodecomposition of rhodopsin. The carotenoid separates from the opsin and breaks down through several intermediaries to colorless all-trans-retinal. The all-trans-retinal regenerates to 11-cis-retinal with the help of vitamin A. This then combines with opsin to form rhodopsin. We shall see all these processes in the next figure so that we have a clearer picture of this visual cycle. This figure shows the visual cycle. On excitation of the retinal receptors by light energy, the rhodopsin, which is present in the receptors, breaks into 11-cis-retinal and opsin. The 11-cis-retinal, through many intermediate processes, changes into all-trans-retinal, which is responsible for the impulse generation. And then in the presence of vitamin A, it changes into 11-cis-retinal, which combines with the opsin and results in regeneration of rhodopsin. So this is the visual cycle which is responsible for the generation of the visual impulse. The above photochemical changes induce electrical responses similar to those in other sensory receptors. They are biphasic and vary in frequency with the intensity of stimulus. Any single nerve fiber reacts when a considerable area of the retina is stimulated. This receptive field, which is about 0.5 to 1 mm, indicates that each ganglion cell is linked with a number of receptors. There are differences in the reaction by stimulation with different wavelengths of light. The electrical impulse, that is the receptor potential thus generated, travels through the retinal nerve fibers, optic nerve, and the visual pathways to reach the visual cortex in about 124 milliseconds. This figure shows the path of the visual impulse. So the visual impulse is generated at the level of the retinal receptors, and then it travels through the retinal fibers to the optic nerve, the chiasma, the optic tract, the lateral geniculate body, the optic radiations, and finally it reaches the visual cortex, which is responsible for the perception of the image. And the time taken for this journey from the retinal receptors to the occipital cortex is about 124 milliseconds. Two kinds of cells have been distinguished throughout the visual pathways: the large magno or M cells and the small parvo or P cells. The magnocellular and parvocellular pathways have different sensitivities and appear to be doing parallel processing of the image. Successive cells in the visual pathway, starting from the photoreceptors, ganglion cells, cells and neurons of the lateral geniculate body, striate and extrastriate cortex, are involved in increasingly complex serial processing of the image. Thus, there is simultaneous, serial, and parallel processing of the image. This figure illustrates the parallel and serial processing. In parallel processing, multiple units or bits of information are processed at the same time over multiple transmission lines, whereas in serial processing, one unit or bit of information is processed at one time over single transmission line. The simultaneous, parallel and serial processing of the visual impulse enables us to fine-tune and appreciate innumerable variations in object color, hues, size, ever-changing location and distance, contrast, contours, background luminance, and many other variations. Further fine-tuning is enabled by the ability of the pupil to change size as per the luminance and distance of the object, the accommodating power of the lens, the tracking, fixation and refixation movements achieved by the extraocular muscles, simultaneous binocular perception, and the ability to use both eyes in coordination. Remember, 6 out of 12 cranial nerves serve the eyes. What are the components of vision that enable us to fine-tune this function? So, if we try to simplify it into components, we would see that there is a light sense, there is a form sense, there is a color sense, and there is contrast sense. We shall look into these briefly to understand what they enable us to do. Light sense is the ability to perceive light and its gradations of intensity. The lowest intensity of light below which it cannot be perceived is called the light minimum. It varies with the amount of light that was initially falling on the retina and the speed with which it is reduced. the dark adaptation. The rods are more sensitive to dim light and cones are more sensitive to bright light. In dusk, we see with our rods, that is called the scotopic vision, and in bright light, we see with our cones, and this is called the photopic vision. Form sense or perception of shape of objects. The cones play a predominant part in this function. The cones are most closely set and most differentiated at the fovea. Ability to distinguish shapes of objects is assessed by visual acuity. Form sense is most acute at the fovea and falls off rapidly towards the periphery. This gradual decline in vision from the fovea to the periphery is represented by the hill of vision. Absence of receptors at the optic disc leads to the blind spot or the non-seeing area in the field of vision. This is shown in figure 5. This figure shows the visual acuity in different parts of the retina or the hill of the vision. So here we see that the tip of the hill, this corresponds to the fovea. Here the vision is the finest and it gradually declines as we move towards the periphery, and this is represented by this hill of the vision. And here, if you see here, there is a sudden decline here. This corresponds to the optic nerve head, and this is called the blind spot. Color sense or the appreciation of colors is the function of cones, hence occurs during photopic vision. Our vision is essentially trichromatic. This is the basis of young Helmholtz theory of color vision. The pigments in the cone absorb preferentially different wavelengths of light, red, green and blue as shown in figure 6. All other colors including white can be perceived by a combination of these three primary colors in different proportions. Here we see the absorption spectrum of visible light by three cone pigments: the blue, the green and the red. The other colors are perceived by various combinations and selective absorption of these three primary colors. Unlike visual acuity, contrast sensitivity is the ability to perceive slight changes in luminance between regions which are not separated by definite borders as shown in figure 7. It may be impaired even in the presence of normal visual acuity and results in mild fogginess of vision. Contrast sensitivity may be affected by many factors like age, refractive errors, amblyopia, glaucoma, lenticular changes, retina and optic nerve diseases. These are the charts used for assessment of visual acuity and contrast sensitivity. This is for the literate patients, these are for illiterate patients, these are for small children, and this is the contrast sensitivity. These charts are here to give you a visual impression of what I mean by visual acuity and what I mean by contrast sensitivity. How we test it, how we record it, we shall discuss in subsequent lectures.